Good afternoon. Thank you for attending this second streaming class of our bioinformatics workshop. As you know, yesterday we successfully kicked off the program. We have seen that you are actively working with the Moodle material, and I'd like to thank you for that. Let's turn our focus to one of the most fundamental software tools in bioinformatics, the basic local alignment search tool, or as you have probably heard more often, BLAST. Since this is a really popular tool applied in many fields of biological data analysis, we find it important that you get a detailed introduction on how to use it properly for finding sequences in the biological databases introduced yesterday. So let's see.
Hello and good morning. Today we are going for the third episode of our bioinformatics workshop, and this time our topic is BLAST searches based on sequence similarity. Please meet my teaching assistant Gaudi, a little one-year-old boy who's been helping me, and he's not going to be in the picture for very long, but anyway, he's here to support me. So this BLAST searching means database searching by sequence similarity. That is the concept we discussed yesterday. And the idea in sequence similarity searching is that you have a sequence of your interest, which would be the query sequence. And the question you want to ask is, which sequences in the database are most similar to my sequence? And the algorithmic way to do that would be to make pairwise sequence alignments with every sequence in the database and sort them by alignment scores, but it's not really feasible. There's too much computation with hundreds of millions of sequences in the databases. So what you need to do is to do a heuristic scan, which is quick to do, to identify top scoring sequences, a few thousand, some tens of thousands of sequences maybe, and then do the full alignments only in the short list, and then sort by the scores and give the output. And BLAST is basically a household term in bioinformatics. It's short for Basic Local Alignment Search Tool, and it was a revolutionary algorithm back in the day. I think the original paper comes from '92. It's so fast and so reliable, and it helps so much in doing bioinformatics research that it's really great. And the good thing is that they've kept up the development, and the algorithm as such is so good that it is still useful. We have the BLAST 2 version from a later date and some modifications called Delta BLAST and Psi BLAST for special purposes, which we'll get to know. So once you do your search with a query sequence, what do you get there? The output is most importantly a list of top hits ranked in the ordering of the score you get from it. And also, as we'll see in the demo, there's a different set of results, which is pairwise alignments of each of the top hits to your query. And there's a graphical output which shows which part of your query, which parts of the hit sequences are matched with your query. And then in this list of hits, we also have accession numbers in the database, which are links to those hit proteins, so we can have a closer look to see what it is. Maybe this Latin name doesn't ring a bell, and you have to see if it's a bird or a fish. In this case, I guess most of you would be guessing it's a crocodile, and you would be correct. Then the sequence descriptions here are links that will take you to the pairwise alignments, And in addition to that, we have some statistics of the hits. We have the length of the sequence that was matched to your sequence, your query sequence. There is something called E-value, which is for statistical significance of your hit. And also there's percentage of identity. And then how large part of the query sequence was covered by the matching sequence that could be aligned. And there's so much more. We have taxonomical reports of the hits. We have a distance tree of the hits automatically generated that we could study if it's a reasonably small amount of hits. Now, when we start running the BLAST algorithm or running the BLAST server, so there are some choices to do. One of these things after you've selected your query sequence obviously is in which database do you want to find your hits. Do you want to find DNA sequences which are similar to your sequence or protein sequences? Do you want to look for all possible sequences in the so-called non-redundant collection as discussed yesterday, or maybe RefSeq? Or if you're interested in structures, maybe you should be targeting your search into the database of protein sequences extracted from the three-dimensional models in PDB. There's also a possibility of limiting your hits by species, so you can do a search in a single species, or you might do a search in any taxonomic division as defined in NCBI taxonomy, which we'll use in one of the demos. And once you decided on your query sequence and database, then you just have to figure out which is the BLAST algorithm. The algorithms depend on the database and the query sequence because you can directly compare proteins to proteins and nucleotide sequences to nucleotide sequences. But then if you're doing a crosswise comparison, proteins to DNA, you have to do the translations. And there are a number of translated versions. tBLAST-N has a protein query sequence and a DNA database, in which case your DNA database would be translated on the fly in all 6 reading frames. There's BLAST-X for the other direction, and there's even tBLAST-X, which compares DNA to DNA, but compares translations in the query sequence and translations in the database. And you might be wondering, what's this talk about 6 reading frames? Your textbook tells you that there are 3 reading frames because we have triplet codons. But you have to consider that your query sequence or the database sequence might be coming from the reverse strands, and in that case you have to take the reverse complement of the sequence and consider the 3 reading frames there. The translated algorithms require a lot more computation. tBLAST-N and BLAST-X would be 6 operations per comparison, and tBLAST-X is 36 operations per comparison. All right, let's go to BLAST and do a demo, a BLAST search, which is targeting alligator and crocodile group of reptiles, and with a query sequence of, if you do this at home or in your office, so feel free to choose anything else, but I will be choosing human carbonic anhydrase 3 as the query sequence. And even with a small group like alligators and crocodiles, there will be more than 100 hits, so I have to increase the number of max hits in the parameters to 500. Otherwise we choose default parameters, and we haven't really talked about parameters, which will come after this first quick demo. And you might want to pay special attention to the filtering step of the results. We'll be getting rid of sequences which are too short by using the query coverage parameter as a filter. And also we'll take a short look at the similarity values, the E-values for significance of similarity, to see if we can make conclusions about the hits, if we can see the difference between autologs and paralogs here, as shortly described previously. All right, we are in the website of NCBI BLAST. This is the link given to you in today's section of Moodle, and we want to use the protein sequence of human C83 and search against protein sequences. So this is normal protein BLAST. And now here we could paste sequences. We can even do this with multiple sequences to get multiple sequence sets, but we are just happy to do this with one sequence for now. We have the human carbonic anhydrase tree, and copy pasting the accession number is good enough as long as it's an accession number from NCBI. And we want to do this in the non-redundant sequences for now, and we have picked up the taxonomical ID which we discovered from the NCBI taxonomy. I searched for alligator, and going up a few levels I realized that the upper level taxon that covers both crocodiles and alligators and also gavials and caimans is called Crocodilia, with an English name not so scientific, alligators and others. But anyway, this is what we have here in the organism filter. These organism filters work nicely so that we can have multiple organisms that are combined, and we can have some organisms that are excluded. So if we wanted to have alligators and others which would not include caimans, we could take out the caimans. But anyway, this is good for now. And then we wanted to go for 500 sequences and restoring the default parameters. You can see that the default is 100 and we want to increase into 500. So this is important. The algorithm parameters are hidden so that the casual users are not using too much resources. They get things done with easy parameters, fairly low sensitivity for the word size and so on. But we just want to go for 500 and talk about the parameters in a minute. And then the button BLAST will be doing the BLAST search. And there we go. We have the results ready and they are here. So the results page gives us details about the search, how we did it, and what was the query length, and there's also some statistics somewhere, but let's skip that part. Now the results start here and we have multiple tabs. The first one, the description, is the one for which you saw the top in the previous presentation. And then we have the graphic summary. This shows what is the quality of each hit. These colors are given by scores in each hit, and the length of the bar matches the sequence of your query sequence. So you can see that there are many that are pretty much the complete length, but then there are some which seem to be fragments which are way shorter than the full length. And then going down in lesser score, less sequence identity, again we have a lot of long sequences, some that are slightly truncated, and so here it is. And we also get as a bonus the matches to the conserved domain database for our query sequence. And again, this is something you've seen yesterday, taken from the NCBI protein page. We have the alignments, but we might want to do the alignments from here. So let's just see what it looks like. We have something that is with a high coverage. Let's say this alligator CA13, 53% identity, 96% query coverage. This is still an excellent alignment. This is clearly homologous. There is a little bit of an insertion in the subject, so the query here is our sequence and subject is the match sequence, the hit sequence from the database. And this midline here shows the alignment in a symbolic fashion. So whenever there is a letter, it means that the query and the target were identical. And then when you have a plus, so it means that you have high similarity between the amino acids in that position. So here we have leucine aligned with valine, and you should know that both of these are medium-sized hydrophobic or non-polar amino acids, non-polar residues. And so the only thing which makes us wonder a little bit is this insertion. Is this relevant? Is this for real, or is this a misprediction of the splice site so that it shouldn't be there? And we can scroll down and up in the alignment lists, and you see that it's pretty much the same fairly good matches there. And scrolling all the way up, we can go back to descriptions. This taxonomy report is kind of boring because we had a limit of crocodile-like organisms, crocodiles and alligators, so we can see that the number of hits in crocodiles is 26, gavials 28, alligator mississippiensis, just one species of alligator, 61 hits. And there's another one, Chinese alligator, 38 hits. It can't really be that two different species of alligator would have such a difference in numbers of CAs. We might think that maybe this is just having so many alternative sequences. But let's look at filtering. I promised to show you that in this demo. So here we can filter by organism, so if we just want to pick the hits of a single species, we can do that. Or maybe we want to filter by identity percentages or the e-values, the significance of the score, which are here, which we'll talk about later. But for now, let's just look at this query coverage. So when we looked at the graphics here, it's obvious that there are many sequences that are kind of short. And if I click on this one, there should be links to the alignments too. Yes, here we go. So this is an isoform, one alternative predicted version of splicing, or a version of a selected start site. And you can see, looking at the numbering here, that this is about 30 residues shorter. And then the next one, which is even shorter, this is 72 shorter than the query sequence, so it's missing a lot in the N-terminus. So we want to go for high coverage so that all of these short sequences would be eliminated. And looking at the results here, so you can see that the query coverage is 70% something. And you can see that many of these are isoforms, isoform X2, X1, and in this case, this isoform X3, just 70%, and the isoform X2, 69%, and there should be a better coverage somewhere. But anyway, we want to get rid of those small coverages, and let's do a filtering to choose 90% of lower limit and 100% of upper limit. And filtering in the results, we had 100 something previously, 137 maybe. And now there are just 102. And if you look at the query coverage, so it shouldn't be surprising that all of them are higher than 90%. And this is a good start in the quality control of the sequences here. Maybe we could go even for 94. Let's see if it's drastically decreasing the sequences. 85 sequences selected. So there's still plenty, and this might be okay if we want to see the best example of each CA isoform in each species of alligators and crocodiles. And then I wanted to draw your attention to these E-values. This is a measure of the significance. The smaller the value, the higher the significance of the match. And this is the exponential notation, so 4 times 10 to the minus 37th is an extremely small number compared to the estimated number of elementary particles in the universe, which would be something like 10 to the 90th. So you would need to take quadrillions, probably like octillions of universes, and then you have a chance of finding one elementary particle in all of them. That is the chance of finding a similarity this good by chance alone. So we are pretty sure that the similarity is not due to the chance, but it is due to the fact that these sequences are homologous. So we can infer homology from a highly significant E-value. And now if you follow the numbers here, so here's a jump of 15 orders of magnitude, and unsurprisingly we have a transition from CA3 into CA2, so we have the orthologs up here, and all of these are probably true CA3s, and then we are going down to CA2s. Now it's getting risky when you go below the orthologs to say anything about the relatedness, but in this case CA3 and CA2 are the two closest paralogs. So it happens that all of the CA2s are not very far from CA3s, and they are closer than any others. But then CA13 and CA1 are just about equally close from CA3 and CA2, and they are very close to each other. And what happens is that due to random factors, we have these 13s and 1s slightly mixed between each other, but it is one group that is fairly unified. And again, the e-values go from 10 to the minus 119 to 10 to the minus 103rd. And then again, there's a jump of 7 orders of magnitude, and we go to CA7, which is a cytoplasmic CA and still fairly close. CA5 is mitochondrial and a little bit further away, and this is testified by another jump in the e-values by 15 orders of magnitude. So this is pretty cool, and then we have CA-related proteins which are quite different but still highly significantly similar, clearly homologous, and we have a jump of 20 orders of magnitude. And below that, it's starting to go into a mishmash fashion, so that we have CA12, CA6, CA14, and whatnot all mixed up. But this gives you an idea of what to look at in these e-values to see if there are any jumps in the e-values, so that you can pick the orthologs, the true orthologs. And we are lucky enough that these were properly annotated, so that the names can help us in seeing if these jumps in e-values have any relationship with real biological relationships, like transitioning from orthologs to paralogs. Now let's go back to the graphical summary of hits, and you can see graphically now that the filtering of hits based on query coverage indeed had an effect, so that we have the N-terminus cleaned up. There are no very short fragments and just tiny bits missing from either end. Let's move on to the second half of this lecture, and we'll be talking about parameters and settings we have in BLAST and 2 special versions of BLAST. In addition to this, we'll have a more organized look at taxonomic filtering we saw in the previous demos as a very quick peek. Now, we've talked about BLAST being a heuristic algorithm, so that it's making some shortcuts and assumptions to come up with the most likely best hits. And the sensitivity of the first heuristic pass is dependent on some parameters, and the more sensitive you make it, the longer it takes. So it's a compromise where you just have to be happy with something that's proven to be good enough. And we'll look at the parameters here in the next few slides. And if you are loosening or tightening your parameters, it might have an effect on your results. If you have set a threshold for the e-values which you intend to find, so with other sets of parameters you might find more or less of sequences that cross the threshold. And likewise, the alignments might look a little bit different. So with a more permissive parameters, the alignments might go to a few residues longer in the ends. But then the price to pay is slowing down the search. You saw the magic happening in the demo so that I had the results immediately, but I confess I was cheating. They were run beforehand with the same parameters, and then I just recorded how to set the run to start and jumped into my second window where the results were waiting. In fact, it would take a minute or several minutes, and with slower parameters it might take like 5 or 10 minutes. But anyway, let's start looking at these things. We have some choices which are pretty obvious, like choosing how many sequences you want listed, and also choosing to filter for taxonomy is pretty obvious as soon as you understand what are species and genera and families in biological taxonomy. But then the parameters we choose for making the alignments for the final scoring and some BLAST-specific parameters which affect the sensitivity are the more important topics we'll have to look at now. Now, in the alignment parameters, one of the important ones is substitution matrix. If we have 20 amino acids, not counting for rarer cases, it will be a 20 by 20 matrix in which each cell gives a value of how many points we give for substituting one amino acid by another, or how many points we give for keeping the amino acid as it is. So basically it is a scoring that gives plus points for matches and minus points for mismatches. But depending on the severity of the mutation, the change in the amino acid, there are some which are less often permitted, and therefore they will be given higher penalties, lower points, more negative points. And this is for scoring of the alignment. And the default matrix we have in BLAST is called Blossom 62. We have a few words on that later. And it's a nice all-around matrix which you should start with. And only when you think that there's a reason to take a stricter or a more relaxed matrix, so it's a good idea to choose something else. And there is one task in the homework which is a good example of a case where actually changing your matrix will improve the results. You will find more related sequences. Then another thing in scoring alignments, we don't really have time to go into the theory of pairwise alignments, but when you do the alignment you have to allow for gaps so that you can simulate insertions and deletions in biological processes. It's not only point mutations which happen during evolution and during the descendants of genomes. And to get those indels in their places, there have to be gaps, basically dash characters, that are inserted in the alignments. And not to make the alignments to be extremely long, consisting of more gaps than sequence characters, you have to give a penalty for the gaps to optimize them to a reasonable amount. And these gap penalties have to be matched with the substitution matrix and the scaling of the substitution matrix you use, and there's no reason for a casual user to touch these gap costs. There's one cost for starting a gap and another one for every additional amino acid or nucleotide added in it. And they change automatically when you change your matrix, so just remember not to touch them, and unless you want to experiment. But for normal use they should be as they are. And this is what a substitution matrix looks like. This is just like the top 7 by 7 corner from the amino acid matrix. And you can see that on the diagonal you have positive points for the amino acid staying the same in case of D to E, that is aspartic acid to glutamic acid. You get positives even when they are changing to each other, which is understandable. They are negatively charged acidic side chains, so they function pretty much the same. And then the numbers of negative points are different. For instance, glycine to alanine is considered a neutral mutation, so that it's not given any punishment in the alignment scoring. And these matrices have been obtained by studying substitutions in actual data derived from aligned protein sequences. And the frequencies of each kind of substitution or conservation event can be converted into odds and logarithmic odds, and in that way these matrices are arrived at and then multiplied with suitable numbers so that you arrive at integers for computational efficiency. But this is a part of Blossom 62. And now the number in Blossom 62 indicates that it has been derived from a set of gapless blocks of alignment in which no sequences have been more than 62% identical to any other. And that has been the data for creating these frequencies of substitutions. And then two other commonly used ones are Blossom 80 and 45, and so when the Blossom 80 has been created from more closely related proteins, you should be using it when you are studying and trying to catch more closely related proteins, when you want the conservation to count for more and the substitutions to be punished more, so you would choose that. And the opposite is true for distantly related proteins. So Blossom 45 is made using distantly related proteins, and the result is that because the dataset has more substitutions happening, there will be less penalties or less penalties, not that high penalties for substitutions, and also there will be less rewards for matches. So this should be enough for you to get a hang of the problem in homework. Now, going to these BLAST-specific parameters, the step of heuristic scanning of all sequences in the database is based on dividing the whole database and your query sequence into words of a specific size of letters. And mathematically these words would be called k-mers, so that, for instance, in default BLAST for proteins, you are studying your sequences as 6-mers or hexamers. And even if it sounds complicated that you are dividing your whole database into hexamers in a tiled fashion so that they are overlapping hexamers, and then making a hash table which includes the positions of each hexamer. So then you are trying to find matches of many hexamers in close proximity to show a conservation in your sequence. And in that way you get your shortlisted version. And the algorithm would be allowing for some mismatches because it's not so common to find exact matches of let's say 12 amino acids in a row. But anyway, for faster but less sensitive procedures, which is the default in BLAST, the word sizes would be 6 for protein and more than 20 for nucleotides. And then for more sensitive and slower runs, you can take word size 2 or 3 for proteins and 16 for nucleotides. Okay, this might be a little bit esoteric, and I guess that many of you would never touch your word size setting, but just so that you know there is such a setting. But then something you should be aware of is the e-values, the expect values, and the expect threshold you set for limiting your list of hits. This expect value is a statistical significance measure for each hit. It's basically telling you what is the expected number of chance matches in a random model to give you the score that is given by your query sequence against that target. And if the e-value is 1, meaning that you expect at least one sequence to be found in a sequence database of the similar size but random content, so then you can think that maybe that is a hit that is easily coming just by chance. And it's nowadays the default expect threshold is 0.05, meaning that the most obvious insignificant hits are cut off, but depending on your sequences, if you have longer sequences, it is quite safe to go to 1 part in a million or 1 time 10 to the minus 6 as the cutoff. You can just type the exp notation here. You don't have to start counting zeros after the decimal point. And I would recommend you to take a look at these 2 videos created by NCBI. They are about 3 minutes each, and you can reach them through the link which is in the BLAST pages, but also I've made these into hyperlinks that take you to the part 1 and part 2 of the tutorial. Okay, now taxonomical filtering. This is something you see as one of the main choices in BLAST. Obviously, the most important thing is choosing a query sequence, and then there's the question of getting your database selected right, and also then filtering for species. And once you set an organism filter, it means that only hits from organisms you chose will be shown. So this filter would mean that you are looking for bacterial sequences only, and anything from eukaryotes or archaea is going to be discarded if such hits would be there. And it's good for simplifying things. You just have to remember that your hit list is not going to be complete. It is just what was found based on your selection here. And this is cool because you can add many taxa here, and they would be searched for as a union, so that if you add bacteria as one line and archaea as one line, so you would get hits from either of those 2 groups. But then you can also add a line and then put a tick mark in the exclude box. It would mean that you are looking for the first group, but not anything that is in the second group, and we'll have an example in the demo. Then, as the final part here in the theory, I'd like to discuss 2 special versions of BLAST which you might find useful in special conditions. Well, one of them is an iterated version of BLAST, meaning that you have to run it several times and the results are developing over the run. This PSI is an acronym for position specific iterated. And the position specific is a reference to the way that the scoring matrix is built. So instead of using the same 20 by 20 scoring matrix for every position in the protein, there is like a 1 by 20 matrix which is different for each position. So for instance, if you have a position in which the hits of the first round show there's a fully conserved histidine, then you would be getting really high plus points for having that histidine there, and pretty steep penalties for having anything else than histidine. But then for a position that is a mix of 6 different amino acids, you would be getting fairly low positive points for having any of those 6, and not that steep penalties for others. So this is a way of increasing the sensitivity of the matrix or the search by having a specialized combination of scoring for separately for every position in your query sequence. But to generate that position-specific matrix, you have to start with a normal BLAST-P search, and after that, when you have chosen SciBLAST, then you have to hit for the iteration in the second round, and you will see that if you have found any new sequences which were not significant in the first pass, and you just repeat a few times until no new sequences come up. Okay, another one which is useful and is a fairly new thing from the last few years, it's something called Delta BLAST, which is short for Domain Enhanced Lookup Time Accelerated BLAST. And in plain English, it just means that they are making use of the domain database, CDD, conserved domain database, which makes it faster. So instead of having to do scoring by the normal alignment procedures, they are finding a match if they find a match in the CDD database, so then the domain definitions, which are basically frequency matrices, can be taken into use as the specific matrix for scoring your potential hits. So in a way, it's like a one-step SciBLAST, but it's dependent on having an appropriate domain definition in the CDD database. So sometimes it works better, sometimes there's just no difference to doing a protein blast, and sometimes it's really poor, so that you get no hits in Delta BLAST, but if you do a normal protein blast, then it's okay again. And I think there was a problem that Delta BLAST and taxonomic filtering did not work together. That was the case a year ago. But anyway, it's advertised to be as sensitive as SciBLAST, but quite a bit faster, so you might want to try that. Now let's move to a second hands-on. So I'll be doing a more complex example of the taxonomy tools and talk about the parameters available in BLAST while they are on the page to give you a kind of a visual memory. And then, if time allows, we'll do a comparison of BLAST-P and Delta BLAST. If I can't do a demo for the lecture, so this is something that will be left for material in the forum. Okay, let's do a demo and set a goal that we want to find out what kinds of alpha carbonic anhydrases we have in chordates, animals which are kind of precursors for vertebrates. And to explain what I mean, let's have a look at NCBI taxonomy. I've added the information of the number of genomes in each group. And so in the chordates, the huge majority of all genomes and all sequence data is in vertebrates. But then we have a couple of groups here that have some genomes. Sea squirts are something that you may have seen attached at the bottom of a boat or on rocks. And this group includes the lancelet or Amphioxus. But anyway, because vertebrates are included in chordates, so just trying to look for chordata would give us pretty much everything. So what we need to do here in BLAST settings, I've added the accession code for human CA3 and chosen refseq, and now we need to choose chordates, and it's here. But so now we would be only looking at sea squirts and lancelets and acorn worms and what not that belong in the non-vertebrate chordate group. And let's look at the BLAST parameters. We can do now a dual function. This will yield us a fairly small amount of sequences because these organisms don't have so many CAs per one species. So we should get less than 100. So let's do an exercise where we do one search with the default parameters, long word size, Blossom 62 matrix, and then try to go into more sensitive by dropping the word size to 3 and to going to Blossom 45. So this would be the first search, which is already done. The second search, word size 3 and Blossom 45, and you may note that the gap costs have been increased a little bit at the same time. Okay, we run a BLAST, which is already done. And then a third search, which we would do. Let's go back to default values, 250 sequences, and choose Delta BLAST. Now, pay attention to the word size. As we choose Delta BLAST, so it's automatically going to 3 here, and 6 is no longer a possibility. That is part of the sensitivity here. And now we have some new parameters which are related to Delta BLAST and Psi BLAST. So if we have the ETH value threshold at 1, so it's making sure that we, or it's suggesting that we wouldn't be taking the very worst sequences into account. So we can have a secondary threshold, but now that they are the same, so all the sequences will be counted there. And now in this Delta BLAST, we have an option of going on with further runs of Psi BLAST, but that's not something we are going to do. And now with these parameters, we would start Delta BLAST. And again, I have chosen this show results in a new window, so I can use this same launching screen for setting up multiple runs and getting the results in different windows. And now let's look at the results. This is with the regular default parameters. We get 39 sequences, and even the lowest ones are pretty good with e-value of 1 in a million. 24% identity, good query coverage. These are 2 from Siona, and the scores are 52 and 68. Going to word size 3 and Blossom 45. So the Blossom 45 allows us to align some more residues and get get a slightly better score because it's not punishing so harshly for mismatches, but only so very slightly. And again, it is the same 39 sequences we get as the result. Just a difference in scoring and slightly better e-values, slightly smaller e-values. And now in Delta Blast, what we get is even better scores and very good e-values for this lowest scoring too. And now, incidentally, the ordering of the last few sequences has changed so that this one has dropped from last but one into the last position out of the 39. But unfortunately, what we got here is not helping us in demonstrating the improvement in Delta Blast, but maybe seeing how these marginally significant sequences got to be scoring quite a bit better. You can imagine that in a case where we do have a protein family that has really distant members, we would be able to pull them from beyond the grave back into the land of living, if you allow the metaphor.